These are the points which I am going to focus upon. Let us begin. So to start with the very basic, mutations are the random changes in the DNA sequences that may be due to the replication error or due to the environmental factors. These mutations can target the regulatory regions in the genome which are responsible for the regulation of gene expression, and such mutations are called the regulatory mutations. Now the question arises, what are actually the regulatory regions? So there are certain regions in our genome which has no power to produce the protein, but they actually have the binding site for certain factors and thus regulate the gene expression. These elements are called cis elements because they are present on the same piece of DNA on which they are regulating the genes expression. The factor which bind to these elements are called as the transregulators because they are not necessarily encoded by the genes present on the same DNA. Now, the cis elements and the transregulator producing gene, they both may undergo mutations, regulatory mutations actually, to cause influence on the gene expression. Now the question arises, what are the nature of regulation? So it may be a positive regulation enhancing the gene expression, and it may be a negative regulation suppressing the gene expression. Here is the picture which is showing that the genome has the power to loop upon itself so that the different cis elements and the bound transregulators can come in close vicinity to the promoter region and the bound RNA polymerase, and thus influencing the gene expression. So we are going to illustrate the lac operon model in order to understand this topic in more detail. So in lac operon, the basic information is that cis elements are promoter, operator, and the cap binding site, and the transregulators are cyclic AMP binding protein and the repressor protein. The very basic phenomenon which we understand here is that lac I gene produces the repressor protein which binds the operator and in turn doesn't allow the binding of the RNA polymerase and promoter, and thus stops the transcription, whereas the catabolite activator protein binds to the CBS or the cap binding site and thus enhances the transcription. When operator, so we are now going to deal with the regulatory mutations which we see in the lac operon. When operator is normal, then the repressor protein produced by lac I gene is coming to bind here successfully. But when the operator is bearing the regulatory mutations, then repressor protein is returned by the operator sequence which is mutated. This mutation is denoted by OC or O superscript C, where C represents the constitutive, means this is promoting the constitutive expression of the genes present on the lac operon. The promoter normally binds the RNA polymerase and enhances the transcription, but when promoter has the regulatory mutations, it returns the RNA polymerase, and thus there is no transcription and no formation of the product. Lac I gene, which produces the repressors, when it is normal, it produces the normal repressor by successful transcription and translation, which binds to the lac O sequence and causes the transcription. But when lac I gene has lac I minus mutation, then in that case the mutant repressor cannot bind the normal lac O plus, and thus there is the successful binding of RNA polymerase on promoter region and the transcription continues. When this lac I gene has the regulatory mutations lac IS, then the proteins which are formed, the repressor proteins which are formed, they are called super repressors. Why? Because they are not allowing the binding of the inducer over them, and hence they successfully bind to the operator region in any situation, whether inducer is present or it is not present. In that case, the transcription is not possible in any case. Lac I gene, which is having lac minus I minus D mutation, that produces such homotetrameric units which are defective and thus cannot bind the operator, and this is also the case where the transcription continues successfully and constitutively. The cap binding site may also bear certain mutations, like if the cap site which binds the cap protein is mutated, then in that case cap protein cannot bind and the transcription cannot happen. Similarly, if the cyclic AMP region, the cyclic AMP is not able to bind to the cap protein due to any mutation in the cap protein, then also this entire complex of cap and cyclic AMP is not going to bind the cap site, and in that case there is no transcription or there is low transcription. Other than this, there are certain spacer sequences between the cap site and operator, which if inserted or deleted for certain nucleotides, then they may cause the uh, failure of the cap effect of cap site over the regulation of transcription, and the transcription becomes weak. So these are the points which we had to address in this topic. Thank you so much.